Okay, finish the gram stains. Now we want to go to acid-fast stains. Okay, and acid-fast stains, again, are used to detect the presence of Mycobacterium. And the two pathogenic ones that we worry about are Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes leprosy. We can all, Nocardia, it doesn't, I mean, it can cause disease, but it's very innocuous. What we're really looking for is, and what you're going to see this, is a sputum culture with an acid fast stain. That is almost 99.9% of the specimens come down to the lab is a sputum culture looking for tuberculosis. Okay. So when we look at these organisms, the difference in mycobacteria, as you can see, we went through the gram positives. There's your peptidoglycan layer, and very thick, and then your LPS for your gram negatives. There is a difference in Mycobacterium. They have an outer membrane and they have these mycolic acids. Okay, slightly different. We'll talk about this in a lecture in greater depth, but they have mycolic acids in their cell walls. So it does make them a little thicker and a little different. Okay, so when we do it, we prepare our slide just like we did with the other one. We usually use staph as a control to just, to see. I've used it to show you the difference. All right. And I'm using Mycobacterium flea, which is found in the ground. Mycobacterium flea is frequently used in the lab because it is not pathogenic to us and we can do that acid fast stain and not have a problem. Okay. So you mix them. All right. So the primary stain with acid fast is carbofusion. It's a fuchsia color. It's a deep fuchsia color. And again, it is positively charged. It will be attracted to the cell wall, the negative charge of the cell wall, but we let it stand for 5 minutes to make sure that it can get in. Okay. Some procedures will actually have you put these slides over steam. We used to have a beaker, we boil the water, we put a little rack, and we put the little slides on there and let them steam. And that opens those cell walls up so that you can get the stain in there. Nowadays, they have a procedure you don't have to do that. Okay, and we don't have a mordant. We don't need one. We go directly into, we rinse that. We will rinse that, and then we decolorize. And the difference with the decolorization is we are decolorizing with acid alcohol. We add an acid to this alcohol. And that's where the name acid fast comes from. Because even though you've added acid, the stain stays in, or the stain stays fast in those cells. And that's where you get the name. So you would flood the slide with acid alcohol for 30 seconds. It will remove the peptidoglycan layer of any gram positives or gram negative bacteria. It will remove it. But because you have the mycolic acid, that will stay in. That carbofusion will stay in. It will not get rinsed out because, again, this is much thicker. Okay, and those mycolic acids are going to hold that stain in. Okay, so then we counter stain. Just if we have any other bacteria in there, we want to pick them up. So we counter stain with methylene blue, which is a nice bright, like lovely little blue, for about 30 seconds. And any gram negatives or gram positives that are there will stain the nice blue color. And then we will rinse it and blot it dry, and just like we saw. And in my lecture with the acid fast, I have a picture of the acid fast bacteria so that you can see them. But the bottom line is we're looking for the Mycobacterium is a bacilli. So we are looking for these lovely little fuchsia colored bacilli. and it is reported out either as an acid-fast stain is no acid-fast bacilli seen or acid-fast bacilli seen. One or the other, just that. Okay, so real quick, 5 minute there in a lab. This would take, we'd be doing it. This would be the only stain we'd be doing for the day. Okay, but you guys, so make sure you watch the videos that I have, and we'll see you.